Thank you for your interest in my poster, which is entitled Development of a Novel Surrogate Blister Fluid Matrix for Flow Cytometry Assay Validation to Support Bioanalysis, which will be presented at the WRIB 2026. My name is Martin Turcotte, and I'm the Global Scientific Director for Flow Cytometry at Altasciences, and I will give you a brief overview of the exciting work we have conducted in partnership with the biotech company Argenx. To be able to develop and validate a bioanalytical flow cytometry assay with fresh samples, we need to have an easy access to a certain quantity of the specific matrix that will be used during the clinical trial. Unfortunately, this is not possible with blister fluid. So for this specific projects, the generation of blister on study followed the cantharidin model, where a drop of 0.1% cantharidin solution is placed on the skin of the volunteers. And during the following 24 hours, there is a formation of a blister, which we can then collect the fluid for further bioanalysis, like flow cytometry. While this approach is non-invasive in a context of a clinical trial, it's not convenient to have access in a bioanalytical laboratory to perform, for example, an assay development or a validation. Thus, we needed to create a surrogate matrix with the objective to be as close as possible to a cantharidin-derived blister fluid. In theory, those blister fluid are mainly composed of 2/3 neutrophils, a quarter of monocytes, and approximately 5% of lymphocytes. We wanted to focus on neutrophils and lymphocytes. So we then used a granulocyte isolation kit, which by itself led to a granulocyte purity of approximately 95%, as shown in Figure 4, Panel A. To retain a certain lymphocyte content, we tried only partially using the granulocyte isolation kit, meaning only doing one round of isolation compared to the recommended 2 rounds of isolation by the manufacturer. However, this created a sample that contained too much debris, which led to data analysis issues, such as difficulties to identify lymphocyte, monocyte, granulocyte subsets, compensation issues, background and beads gating, as shown in Figure 4, Panel B. We quickly dismissed this approach and instead focused on the spiking of freshly isolated autologous PBMC into the granulocyte isolate, as shown in Panel C and D. From a quantitative perspective, the granulocyte isolation with 20% PBMC showed the closest lymphocyte content compared to the published theoretical blister fluid, as shown in the last 2 columns of Table 2. So this specific blister fluid surrogate matrix got generated 12 independent times during a fit-for-purpose validation of this specific flow cytometry assay. The T cells, neutrophils, and eosinophils content of those samples are listed in Table 3. We also had the opportunity to add 2 cantharidin-induced fresh blister fluid analysis into this fit-for-purpose validation. Doing so, we demonstrated that the relevant cellular contents were a fit within the surrogate matrix established range. In conclusion, this innovative surrogate matrix approach facilitated the development and fit-for-purpose validation of a flow cytometry assay for regulated bioanalysis, thus expanding analytical capabilities to study local inflammation in clinical trials, leveraging the cantharidin model. This flow cytometry assay showcased in this poster supported the clinical trial earlier in 2025. Thank you for listening.